Log on, tune in, find out. Another good idea from Cambridge.
Okay, so let's see if it works. Okay, so let's start where we left off back in September 2007, so 3-4 years ago. Maybe I didn't have such much gray hair, a little shorter. But at that time, I was giving a lecture about three-partition phylogenetic networks. And I must say, first of all, I had to thank especially Mike and Vincent because they trust me on giving that lecture, although I haven't published anything about that at the time. And I would say 3 years later, I haven't published a paper about that, a single journal paper. There were several ones, so let's say 4 papers. Some of them came up with directly from work done during people I met here at the Institute. Well, I'm lying, it was not 8, it was not 4, 7 papers, 7 journal papers, no, it was 10, no, it was 13 and then 14th coming up now. So it was really very successful and a lot of work. So let me just summarize what we did in these 14 papers. Okay, no, just forget it. Let's get back to the topic of the talk. It will be something different, but also at the end it will be a little related to networks. And I think it fits in phylogenetics, so we'll be talking more about classification trees. I will give a brief overview of taxonomic classification, and then also review some methods used in genomics and in metagenomics for classifying sequences, and especially composition and alignment-based methods. And then the core of the talk will be some new methods for the classification of ambiguous sequences, the notions of taxonomic diversity to compare alternative classifications, and this will be all this big part about taxonomic classification in theory. And then I will show some case studies to see the meaning of taxonomic classification in practical, in real data sets. So to start with, taxonomic classification was already known to the Greek philosophers, and there have been many attempts to classify living things. And maybe the first written account dates back to the early 18th century, Carl Linnaeus, and he studied mostly animals and plants. Well, he also classified minerals. Didn't deal with prokaryotes, but it was really the first attempt to build taxonomic trees. Although as you see in this type, it doesn't look like a tree. It's mostly in tabular form. But it's interesting that at that time this is a very big book, very thick pages, and at that time it was perhaps the first ever book with written in such a typed in a tabular form. Let's see, mathematical audience. So how many of you are familiar with these letters? 1, 2, 3, 4. That's good. It's a good ratio. So Kim Philip came over for green soup. Well, you know, there are several variants of this. Some of them are not politically correct, especially if the talk is being recorded. But anyway, this is a reminder for the standard ranks of taxonomic classification: kingdom or domain, phylum, class, order, family, genus, species. And back to Linnaeus here. Before he introduced actually at least 2 of these ranks, the genera and species, because the names used to at that time to name species were really very hard to remember, very long Latin names. Like, for instance, here, tomato, Solanum, caule, inermi, herbaceo, foliage, etc. So who can remember that? So he kind of simplified and systematized nomenclature, especially adopting this, genus and species. This is called the binomial system of taxonomic classification, and it's due to Linnaeus. And he did a lot of work of classifying thousands of animals and plants in these books. But the real tree, the very first depiction of a taxonomy tree, is due to Charles Darwin. So this well-known manuscript of him, one of his letters. And but then we come. There were several further attempts to come across a exhaustive classification of all living things. Next one, there were several ones. I might be omitting some important ones also, but Ernst Haeckel also provided a classification of introducing prokaryotes in the picture. So we have here animals, plants, and protista called at that time. Then we have more recently Copeland, Herbert Copeland, who refined the picture a little more, and then you have here the Monera, which are prokaryotes. Then we have Whittaker, also refining a little more the picture by introducing fungi. And this is a summary of all these classifications, along with all these classifications were made essentially by observing things. And this classification is a more recent one by Carl Welse, and is due to molecular techniques. So this more or less well established now and accepted, although there's still some debate, accepted classification in where prokaryotes are split into bacteria and archaea, and then we have the whole domain of eukarya with all the previous divisions that were studied, like between animals and plants, and also small eukaryotes. Okay, so you can even find these classifications on the iPhone. I actually wanted to give the talk using the iPhone only, but it didn't work. It worked in the morning, early in the morning, but now it's too late, and the iPhone is as tired as me, so it didn't work. Nevertheless, there are apps like this Taxonomy, where you can query the whole tree of life and search for terms or browse the classification. And I promise it really works. So then there are several methods used to obtain these taxonomic classifications. And first of all, the linear classification methods. Actually, they were not as fine-grained as they are today. So especially Linnean was focusing on the last two ranks, on genus and species for nomenclature, and also trying to divide into kingdom, class, order, and family. So essentially, for animals and plants, he used only class, order, and family, not phylum. And but then there are essentially three kind of methods. So the phonetic methods, or also called numerical taxonomy, that are based on morphological similarity to classify the species. And then the cladistic methods that are based on shared ancestry, so essentially phylogenetic methods that try to classify organisms into clades, which are monophyletic groups. And then there's evolutionary taxonomy methods, which try to combine or integrate the two views. This is all solved in Dan Gatsfield's book, for instance. Can I go on? Okay, but then this was most of this was done before molecular methods. So the only way to classify things was essentially to observe some external traits. And then we have in evolutionary taxonomy methods, they come up with the advent of molecular techniques. And in a sense, this PLO's biology paper, mathematics is biology next microscope, only better. And biology is mathematics next physics, only better. So trying to bridge bring together the fields. But actually, what I think is that okay, we're interested in mathematics here, but also these sequencing technologies are actually biology's next microscope or current microscope, because they allow to look into the sequences of the organisms, and then all the methods we deal with in phylogenetics are essentially classification methods, or some of them are classification methods based on the sequences. And this is particularly relevant with high throughput sequencing technologies that have brought all these technologies to real use by lowering costs and improving accuracy and especially read length. This survey of some of the instruments available nowadays or plan to be available in the near future. And here you see the pictures, and at the end you see what might be coming up later. So an iPhone sequencer or an iPhone interface to a sequencer. So in a sense, we can expect sometime in the future to have a real powerful microscope that will show us the actual DNA sequence. So here you have applied biosystems, Roche, Illumina. Well, the first one is a modern version of the Sanger sequencer, Roche 454, Illumina Solexa, applied biosystems Solid, and then two new coming in technologies, so Helicos and Passive Biosystems, which promise to give a very high throughput, or the order of terabases in a single sequencing run, with a read length goes down until to very short read lengths, and then the last one promises to bring back a very long read length. Turnaround time, so the time it takes to sequence a sample, is increasing, so it can be a whole week for the poor machine working to sequence a single sample. And the accuracy is, I would say, it's comparable. So these figures are not really, they're not available at the time I wrote this slide for the last two technologies, because they are still in testing phase. But these accuracy ratios are not really directly comparable, but are roughly the same. So these Q20 and Q30 are some technicalities, are not exactly the same, but still you see that there's a relatively high accuracy. So this shows a tendency to higher throughput, but a slower turnaround and a slightly lower accuracy. Okay. So now with these methods we came to this molecular phylogeny, or classification of species, due to essentially to Carl West, who had the insight that there must be something special to the ribosome that is being so important for across all the species, that should be highly conserved, so the whole ribosome machinery. So he focused on the study of these particular ribosomal genes, or general ribosomal genes, to look for good markers. And he found out that the 16A in prokaryotes and 18A in eukaryotes was the most highly conserved one across all species. And with this idea, he sequenced 16A's and obtained phylogenetic trees like this one, where you see this is from one of his first papers, 1987. Then at that time he proposed, before him there was only the distinction between prokaryotes and eukaryotes, and he introduced a further distinction between bacteria and archaea. But at that time it was a very new concept, so not everybody agreed on the existence of a third domain or third kingdom of archaea. So he called them archaea bacteria at the beginning, and eubacteria, the proper bacteria. And then later on he, in PNA's paper in 1990, he came back to bacteria and archaea, and they are nowadays accepted names for the species, for these kingdoms. And another key player here is Norman Pace, who further developed these 16A RNA methods, and also developed techniques to isolate very hard to isolate microbes, especially archaea living in very extreme environments. So he managed to replicate these conditions in the lab and grow these bacteria and sequence them. These archaea and sequence them. And this is a more recent picture of the classification of species, where you see the clear distinction between bacteria, archaea, and eukaryotes. And as you see, the difference here is, well, still here, both trees are not rooted. So that's still another point in strong debate, what came first, bacteria or archaea. Okay, so now in genomics there's a lot of work on classification, especially when it comes to prokaryotes, and in particular there's this Berge's manual, which has evolved through a lot of editions. Now it's a 9th edition, and also the manual of systematic bacteriology, which also evolved, and there are this volume 4, there are up to 7 volumes coming up later next year. And based on well, this is a very well detailed account of all known microbial species, prokaryotic species, with not only with the we're more interested in the taxonomy, but also a very detailed account of all the morphological traits. And this Berge manual has been the basis of several taxonomy classifications. Well, on the one hand, we have the NCBI taxonomy, which is a very general repository. It's not necessarily phylogenetic information therein, but it's also widely used. And based on the Berge's manual, we have ARB Silva, Green Genes, Ribosomal Database Project, RDP, and TOBA, the taxonomic outline of bacteria and archaea. So I give you here some web addresses, some pointers where you can access these databases. So some of them are available as web servers, others are available for download. And then there's also more kind of encyclopedic information about these classifications, like the ITIS, Integrated Taxonomic Information System, the Encyclopedia of Life, and the Tree of Life, where some of these databases are also linked to these encyclopedias, and in particular the iPhone application I talked about is linked to the Tree of Life, so you can get a picture and further information of your favorite bacteria. And the same picture comes out more recently in metagenomics, so where we focus now on the study of bacteria, microbial communities living in an environment together with a host. It could be human, could be other host, and it's a very new branch of that affects also phylogenetics, and it came about maybe 10 years ago or less. And this book is a very interesting account of the whole field of metagenomics and the new science of metagenomics. And then there are increasingly many papers in metagenomics in the main biological journals. And there's, for instance, a collection of papers in Nature about metagenomics. And again, what you see here, what we are interested in the metagenomic study of taxonomic classification is instead of a classification of all living things, we are trying to use the knowledge of these taxonomic classifications to to provide an account for all the species and their abundances and other characteristics that are present in a sample of a population, in a metagenomic sample. That could be all the species that are present in all the organisms that are present in a single drop of ocean water, or in a gram of agricultural soil, or in your skin, whatever. So this particular example here is from a paper by Daniel Hudson and others, and was obtained using one of his tools I will be talking about later, which is called MIGAN, for metagenomic analysis. And so you get here a partial tree of species, so classification of all the species that are present in a given sample. In this case, in this particular case, it's a sample of mammoth bone and all the surviving species. And you see here the difference with the previous phylogenetic classifications is that you have this name on the internal nodes. So these are the higher taxonomy ranks that are already known, but also you have thicker dots sometimes inside, which represent the relative abundance of this particular clade, in this case, in the sample you studied. So, and this comes out from aligning sequences, aligning short fragments of a metagenomic sample after sequencing, like the ones you see here. They are aligned to leaves of the taxonomic tree, so to particular species or strains in this case. And then there is some ambiguity here, because when you see in a sample these three sequence fragments, you can assign them to this one, to these three, but this one could be aligned equally well to this or to this bacterial strain. So you're not sure which of them is it. So, and to resolve this ambiguity, what they used in Megan was an LCA, lowest common ancestor algorithm. So just a consensus sequence, so kind of consensus sequence. So just align them to a higher rank, like in this case here. And this how these thicker dots come into play. And we will discuss later methods of improving this kind of LCA assignment. Okay? So there are essentially two ways to do this classification, or actually there are two ways to map the sequencing reads to species in a taxonomy, given full knowledge of the taxonomy and the sequence behind that. The first distinction, well, we were talking first about classification methods. This, sorry, about composition-based classification methods. So they are in a sense alignment-free methods. So they take, for instance, the K-mer composition of the sequences into account to do the alignment. So it's not an alignment in the usual sense that you see. You can visualize each base pair to which base pair corresponds. But still, it can give you a very good estimate of the similarity between a fragment and a sequence. That might be enough, fine-grained enough to allow you for a good classification of the sequencing reads in a metagenomic sample. So the method is basically K-mer searching, although there are several other methods, alignment-free methods that could be used here. And these are the two important concepts here in classification. So what we understand by top hit, because there will be necessarily can be ambiguities, and what's the best stratum. So what we consider as equivalent candidates to identify or to assign or to align a sequence to. So at top hit, you could take the closest sequence. So it seems reasonable to consider the closest sequence in your taxonomy to the sequence read you are trying to identify, where closest here means under some particular metric. And again, there's a large literature on different metrics for alignment-free sequence analysis. And in case of ambiguities, it also seems natural to consider as a best stratum of candidate sequences all those sequences that are at the same distance as the closest one to the fragment. So, but this is when you classify whole genomes. So when all these sequencing technologies give relatively shorter or longer, but they are still short fragments of DNA that then need to be assembled to get the whole genome. Especially in for eukaryotes, this could be very hard task because of a lot of factors that affect the way all these millions or billions of small fragments can be assembled together in a whole sequence with some degree of certainty. But in the case of prokaryotes, which have much shorter genomes, this is more viable here, and then there are many more whole genomes known for prokaryotes than for eukaryotes. But as we said before, we can focus also the classification based on a marker gene like 16A ribosomal RNA. And in this case, the picture is still the same. So no matter we classify, we have as input to the classification problem whole genomes coming from this metagenomic sample being sequenced, or 16A ribosomal RNA sequences. The definitions are the same, so the methods are the same for composition-based analysis. The top hit and the base stratum are the same concepts. The only thing to mention here is that even in the case of a single marker gene, the structure of this particular gene is also very interesting, and it's not used, so it's not the whole gene that is the marker here, but only a part of the gene. So the gene has an intertwining structure of conserved and non-conserved regions. So the non-conserved regions are the ones that are interesting because they give the variation, they can fit all the species, and the conserved regions serve as primers for PCR. So this is the way. How instead of the whole gene, just one hypervariable region is used for sequencing. So you can see in some paper maybe this is the B4 region of the 16S gene. So this kind of thing. Just think that it's a small piece of 2,000-3,000 base pair long gene. Here, just to give you the literature is huge in this area. So just to give you a point to start with, there's a nice review of alignment-free methods for sequence comparison in this paper in bioinformatics. But this is just one of the ideas. So composition based. So the other one we are. And here you have some pointers of tools for composition-based taxonomic classification, and web servers supporting these tools or similar tools. But we are perhaps more familiar with alignment-based methods. So for whole genomes, because of the many different evolutionary events that might have occurred, it's more appropriate to use tools like BLAST for the alignment of short fragments to long whole genomes, and so that's why the preferred method here is BLAST alignment of sequence reads to the sequences in the taxonomy. And a stop hit, the default value to to take just one value. The default one in BLAST is an e-value of less than or equal to 0.001. And the best stratum, again, is all the sequences with the same e-value as the top hit. So, and this gives, essentially this gives for each read a set of candidate sequences in the best stratum, which are all in principle equally good, equal candidates for identification of this read, and then we need some method to resolve these ambiguities. When it comes to 16S ribosomal RNA sequences, the situation is different because then we have shorter or longer, but essentially short reads that we need to align to short genes or pieces of genes. So the preferred tools are based on the Barrows-Wheeler transform, so based on indexing techniques on suffix arrays, and there are plenty of them available. This is also a well-studied topic in computer science. And now instead of e-value, the measure is based on the number of mismatches in the alignment. So all the sequences with the most k mismatches are, so the top hit will be that, but it doesn't need to be unique. So we have again a best stratum of all the sequences with the same number of mismatches as the top hit. Okay. And again, there are a lot of here's a survey of these sequence alignment algorithms in the case of these new technologies of short and long sequencing reads in briefings in bioinformatics. And there are also several tools available, standalone programs like Karma and Megan, and also web servers like Camera, Galaxy, and MGRAS, which provides pipelines for this and other analysis of metagenomic data. I might be forgetting some, so it's just to give you some pointers here. Okay, so let's focus a little now on this assignment problem. So what we have is, just to formalize a little more, we have a set of sequences and a taxonomic reference classification, essentially a tree with the leaves labeled by these sequences. But it's a classification tree. We also have information on all the internal taxonomy ranks. And we have a set of short or long sequence reads which come from a metagenomic sample after sequencing using one of these technologies I mentioned before, and a parameter, positive inter k. And this is the input to the taxonomic assignment problem. So the output is, for each read, a single node in the taxonomy that represents it, that represents in some good way the subset of candidate sequence I told about before. So all these hits or matches, all this subset of the leaves that contain this read as a substring with at most k mismatches. Okay. So this is essentially for this setting here, but it could still be applied to the previous one. Okay, so now we're clear about the problem. So we have let's say we have we have billions of reads out from 1 or 7 days of working of this poor sequencing machine, and we want to make sense of that. So for each of these reads, we want to assign it, identify it, so where it comes from. So assign it to some species in our known taxonomy, but not maybe because of ambiguities, we might not be sure to which species it belongs to. So we might have several hits or candidates. So let's say we paint with blue these leaves of the taxonomy, and then we can go up and go to the LCA and say, okay, we are not sure about what particular organism it belongs to, but maybe we can tell for sure that it belongs to this class or to this phylum. So this is the idea of taxonomic assignment. Now, how can we measure the accuracy of this assignment? So let's get back to precision and recall. So this just to review the definition, so I will skip it. Precision and recall in terms of true positives and false positives and negatives. There are several ways to combine precision and recall in a single measure. One of the most widely used is the error C curve, or area and the ROC curve. But another one that's very interesting also because it has a nice mathematical foundation is the F measure, which was introduced in the context of information science or information retrieval. And it's just the harmonic mean of precision and recall. So here you have the formula, harmonic mean of two numbers. So it's twice precision and recall over precision plus recall. And the interest of this F measure is that it gives you in a single number, which then are easier to compare, the combined precision and recall of some experiment. In this case, it will be taxonomic assignment experiment. So how would you measure these true positives and true negatives in this context? So let's say we have the ith read, and all the MI will be capital MI will be all the leaves of the taxonomy tree that are candidates for this read, that to which the read can be aligned with up to k mismatches. And then we have the whole subtree of the taxonomy rooted at the LCA of these matching leaves, okay? And then we have, this will be then matches, and NI will be mismatches. All the rest of the leaves of this subtree that are not matches, that are not candidates. So this will be essentially be the measure of the error. But now for this ith read, we can partition the leaves of the taxonomy tree in 4 sets, which will be true positives, false positives, true negatives, and false negatives. So this all the matching leaves are true positives, and all the non-matching leaves are true negatives. And since we are restricting here the analysis to TI, we only have matches and non-matches. So the true and false negatives will be empty here. So the situation looks something like this. So this is our taxonomy, but now we focus on the ith read, and this is the LCA of the matching leaves. These are the matching leaves. So this is the LCA, this is the whole subtree this y, and these here are the non-matches. Of course, this is just a simplified representation. This doesn't have to be continuous here in any sense. So these are here true positives and false positives. And this is a situation when we assign, when we identify a read to the LCA of these candidate sequences. So in this sense, it's easy to compute the F measure, and then we have a measure of precision and recall, a measure of the accuracy of the assignment. And of course, if we assign to some higher taxonomy rank than this one here, we will have an additional error, so it doesn't make any sense. But the question is, what happens if we assign to some lower rank inside this sub-taxonomy? So then let's say now it's get a little clumsier. Now we have the ith read assigned to the jth node in this subtree, in this subtaxonomy. And then we have Tij, the whole subtree, rooted at this node j. Mij and Nij will be the matches and non-matches, but within this smaller subtree. And now it's when we can define true negatives and false negatives. So true negatives are the negatives that are not false negatives before, that were not before, and false negatives are the matches that were not the matches now, that the matches before that are not matches here. So maybe it's easier to see that in this picture. So now we have the whole taxonomy, the LCA of all these matching leaves, Mi, and now we consider this jth node and this subtree. And this is where we have now the true positives and the false positives for this particular node, internal node here j. Okay. And now we have the 4 things we needed to compute the F measure. So here we have another example. If instead we have this painted in red now, these 4 candidates to which the read, where the ith read in the sample can be assigned to, and I is here. This is the root of the LCA. This is the LCA of these matching leaves in this taxonomy. So this is the whole subtree, and now we consider this other. We want to go down one level, one rank, and see what happens with the accuracy. So here we have this subtree ij, and now this becomes a false positive, and these 3 become true negatives for this particular assignment. And we can measure this for each internal node in this tree, and just keep the best, report the best hit as the top hit for the assignment. So now it's the same formulas apply for precision, recall, and F measure, only that we have ij, and we want to maximize the F measure over j for all internal node j's in the subtree rooted I. Okay. So this concrete example, this is a piece of the tree of life, from bacteria down to some marked in blue here matches, and this is the LCA aquaficales of all these matches, and now we can compute precision and recall, and combining in the F measure is 60%. But if we go down, this is the LCA here in red, and here if we go down one rank and consider this one, we are discarding 3. We are reducing the coverage, but increasing the accuracy, or increasing the sensitivity. So increasing the specificity. So we get we increase the precision to 100%, because then we cover all the hits in this subtree, but we decrease the recall from all to half of them, so to 50%. But still, the F measure gives a better result, 67% in this case. So we would prefer, in this sense, this assignment. Oh, so I think I have to rush very much now. Otherwise, we won't have lunch today. So this is the F measure, and we can compute it efficiently. After all this sequencing and alignment steps, it only takes linear time linear in the size of the taxonomy or the subtaxonomy to do all these calculations. But we can go a little further and see generalize a little the F measure. If we see the original formula of the F measure, we can restate it in terms of true positives, false positives, and false negatives only. And then we can generalize it in this way by introducing in a parametric way. So we can introduce a parameter q and 1 minus q, affecting the ratios of false negatives and false positives to true positives. And the good thing is that this is a proper generalization of the F measure, that in the case of a parameter 0, so we only take this side, we are enforcing the assignment to leaves in the taxonomy. If we go, if we neglect the second part and we set q equals to 1, we enforce assignment to the LCA, and in between there's a whole range of possibilities. But in particular for 0.5, we will assign to the F measure, actually to some node in the taxonomy that maximizes the F measure. So in this sense, it's a generalization. So the proof is very simple. Well, it's more or less simple, just manipulation of this formula. So actually the node that minimizes the penalty score for Q 0.5 is the same node that would maximize the F measure. Of course, the node is not unique. I should say a node here. And now, now there are some results about algorithmic complexity. So it's all linear time algorithms. Once we have done the mapping or assignment of the reads to leaves in the taxonomy, to sequences in the taxonomy. And there are two results. One is linear time in the size of the whole taxonomy, and the other one rooted at the LCA. Another one is linear time in the size of the leaves, matching leaves, after some pre-processing of the whole taxonomy. Okay, I will skip this. And this is all implemented in a PER tool, and there's also a C++ version coming up called Tango, which is taxonomic assignment in metagenomics. Now, to compare alternative assignments or taxonomic classifications of a sample of metagenomic samples, there are a lot of ecological indices that were developed between in ecology, especially in microbial ecology, and some of them are listed here. So they are called alpha diversity or beta diversity indices. There are some more until omega diversity, and also richness. So richness is just the number of species in a sample. It's also some complementary quantitative measure. These definitions are, some of them were introduced by Whittaker, and especially alpha diversity is interesting because in this setting it's a measure of spatial diversity within an ecosystem, and here the ecosystem is a metagenomic sample that we are studying, and also omega diversity, which is the difference in terms of phylogeny between the species in an ecosystem. And then it's interesting to take both into account, also to use the distance between species and not just the diversity, but the real phylogenetic diversity. And there are, again, several indices, many indices proposed for each of them. Here we have just the one that was best known or most widely used. For alpha diversity is the Shannon-Wiener index, which is just the information entropy of a distribution taken from a population. So here we have the abundances of species, so Ni, and Pi is the proportion of relative abundance or probability if you want, because N is here the number of species. So it's just minus sum of product of probabilities and logarithm of probabilities. It was introduced by Shannon in 1948 and then further developed. I think it was introduced by Shannon, but he was inspired by work by Norbert Wiener in cybernetics, so that's why it's called the Shannon-Wiener index, but they never published it together. And for omega diversity, there's one of the best known, perhaps one of the best, the first measure proposed was the Clark-Warwick index, which can be understood as a quadratic information entropy, where we have this product of probabilities, and also a weight, which could be some measure of similarity between the species, but in this case it's the distance in the taxonomic reference between the species. So for instance, here we have a TOEIC sample. In the metagenomic sample we have this in the, once we have the complete information, if we have it, then we know that these individuals here we sample belong to the species and so on. And this is the whole metagenomic sample. This would be reads here. And here we have the proportions of probabilities. And this is the value computed here for the, in this case, for the over time. There are some bounds on these numbers, some practical and some theoretical bounds, but essentially it ranges until 3.5, until the logarithm of the number of species.
You ran out of battery.
I really ran out of batteries. So, okay, this index is also between 3.5 and 7.5 in practice. But at least it gives you a single number, a way to compare taxonomic classifications in terms of diversity, or two alternative ways. And I promised just to finish some case studies. I will omit some details there on papers, on published papers, all of them. So the first case study is a metagenomic data set of Roche 454 sequence reads, which are considered to be long reads of 16A ribosomal RNA. And there are several data sets going from human to chicken, rat, marine samples, also marine samples here, and simulated data set using the MetaSim program by Daniel Hassan. And using as reference taxonomy, type cultures, that is where we have more certainty on the sequences, so high quality, near full length, so whole 16A ribosomal RNA for about 5,000 microbial genomes, coming from this TOBA taxonomic outline of bacteria and archaea, with the alignment made by BLAST and also by a tool called GEM, which is based on Barros-Wheeler transform, so genomic multi-tool. Ambiguities are considered to be all the sequences with at most 2 mismatches, which is about 99% sequence identity for this length of reads. And the assignment of ambiguous sequences being made with the LCA algorithm of Megan, and also with this Tango tool, and measuring diversity in terms of Shannon-Wiener index. And okay, so let's say very quickly, this just I promise to give all this out on the web. So this for each of these data sets studied there, the number of hits of sequences not mapped at all within these 2 mismatches, the number of unique sequences or number of ambiguous sequences in the metagenomic data set when using this GEM tool and when using BLAST. And then there's a second study with marine samples of Priest-Post Lake, which have quality information associated to the reads, so sequencing quality information. And we studied how the use of this information influences the assignment. And then you see that when, well, for this reference taxonomy and also for a larger reference taxonomy of over 300,000 sequences, that the number of no hits reduces and the number of one and ambiguous sequences increases. So it's more accurate to use this quality information in the alignment or in the mapping phase here. And these are some results for FAST-A and FAST-Q, so with quality measures, of the number of reads assigned to the different taxonomic ranks as a function of the Q parameter. So that you see ranging from 0, we assign to species, and 1 we assign to the LCA, which can be in any of these ranks. And here this will be the F measure. And so you see there's a smooth increase, move up in the taxonomic ranks when we go from 0 up to 1 for this Q parameter. And the same happens here when using quality measures. So the second case study is for both Roche 454 Illumina Solexa, so long and short reads, and simulated using MetaSim, using the same reference taxonomy as before. But for alignment we use for 454 BWA SW Smith-Waterman and Blatt, and for Illumina Solexa we use BWA and GEM. They're all tools based on Barros-Wheeler transform, but Blatt is a more faster version of BLAST. And then again 2 or 3 mismatches depending on the read length for as a measure of ambiguity, and we use different tools, so Mother, RDP classifier, and Tango for the taxonomic assignment, and measure both Shannon-Wiener and Clark, so alpha and beta diversity. Okay, so in this case it's a simulated data set, so we can measure the real taxonomic diversity. So real, this is the value of the Shannon-Wiener index. This is the number of species, or the value of richness, and this is the taxonomic diversity of Clark-Warwick. So this is a good, interesting experiment, because then we can know how far we are from the actual diversity, or taxonomic diversity. And okay, this is a profile of the metagenomic data set, but this is the interesting result. Well, not yet. This one. So here we measure with. So we have with the RDP classifier, we have the diversity on richness as a function of some parameter, which is a confidence threshold. We tried something more, so I put just some of the values in a table here. So we have for the 454 and Solexa, Illumina Solexa, we have simulated two data sets of the more usual read length, so 100 and 250 base pairs for Roche, and 50 or 75 for Solexa. And we have here the diversity and the richness computed, that we can compare to the actual values of the data set. And the same with mother, diversity and richness. And for Tango, depending on how we do the alignment, if we do it with BWA or with GIM, we get different values of, here we do it only for Illumina Solexa, and we measure diversity, richness, and taxonomic diversity. And in any case, we get much closer than with the other tools, and also with GEM and Tango, much closer to the actual taxonomic diversity. And the third study is just to finish, and then we can go for lunch. Here we have Sanger sequences. So just to complement, to give you the whole picture, so we have Roche 454, Illumina Solexa, and now Sanger sequences from a data set collected by Mavromatis and others. This is a Nature paper, and using this came out of 113 microbial genomes, which were then sampled to produce this data set. So this the taxonomy is the NCBI taxonomy for these genomes. Then we align it in BLAST and take as ambiguities the standard default value for BLAST, and we assign taxonomically with LCA and Tango, and measure taxonomic diversity, so omega diversity. And so there are three different data sets depending on the some characteristics, so low, medium, or high complexity, depending. So let me go back for a while here. So in low complexity, we have one abundant organism flanked by some more or less abundant ones. Here we have several dominant populations, and here there's no, in the high complexity, there's no dominant one. And this reflects the characteristics of different types of metagenomic samples or environments. So it covers more or less the whole picture. And when using LCA or using DF measure Tango assignment, in this case we get this picture of the distribution of the reads across taxonomy ranks. And then we can measure again the omega diversity, and we get, using LCA, these values for each of the three data sets. With the F measure we got better values, and this is the actual value, so it's getting closer to the actual value, but not yet there, because there's ambiguities and it's incomplete information. Okay, so just a brief outlook and then we finish. One open problem that I think it will be interesting, it's already very interesting, is the use of multiple reference taxonomies, because in biology there's not much agreement as to what's the right taxonomy, and so we should be able to use to take conflicting taxonomies into account in this analysis. And also moving on to reference phylogenetic networks, which were the topic of first and yesterday's talks. So We already did some work on in the context of phylogenetic networks about LCA algorithms, and there's also a recent paper came out about LCA problems for DAGs, so which could be the starting point for this. Okay, so most of it is joint work with a new PhD student, Daniel Alonso, and then a former PhD student, Jose Clemente, who's now in postdoc in Rob Knight's lab in Colorado. Jesper Jansson, who's postdoc in Tokyo, Japan, and Paulo Ribeca is a group leader in algorithms in National Center for Genomic Research in Barcelona. And okay, so they cancel out of a few papers. So one of them is a PSP paper last year, and this year a journal version for BNC Bioinformatics. Then there's a Briefings in Bioinformatics paper about all this comparison of classifications, and something we just submitted and to in the Net Journal, taxonomic assignment in metagenomics with Tango. And so you know that Tango then stands for taxonomic assignment in metagenomics, but it also stands for what? Argentinian tango. Okay, thank you very much.
Thank you very much. Maybe one quick question.
What I want in the reference taxonomy, there are usually many errors. I mean, the sequences have been assigned the wrong name. And that could be. I mean, that could be. What do you? I mean, how sensitive are those measures to such things? Do you have an idea?
Well, that was the point of putting this as an open problem using multiple reference taxonomies, because the reason why there's conflicting information.
I don't mean that. I mean just that the sequences are misidentified, or they have been. I mean, there have been errors in the lab. That could be really big errors sometimes.
That's certainly affecting every step of the metagenomic analysis.
Yeah.
Here we are trying to deal, assuming we have the best information we can get before the alignment phase, to do the taxonomic assignment based on this information, and we try to get as good as we can for the taxonomic assignment. But okay, there are many other problems before we get to that, even with sequencing. So there's now recent work about alignment, not from the DNA sequences, but straight from the flow diagrams produced by the sequencing machines, because they claim to be more accurate than the peak calls made by the sequencer programs. And there are many other problems. So of course there's a lot of uncertainty, a lot of incomplete information. But here, at least for taxonomic assignment, we do the best we can with the information we have.
Okay, and I guess the rest of the discussion we should move to lunch, and we'll meet back here at 2 o'clock.